Hello, everyone. Today I'm going to present a paper that was published almost a decade ago in Cancer Research by a multi-institutional group led by Ravi Salgia et al. I hope I'm saying that correctly. This manuscript characterizes MET expression and functional activity and mutations seen in tumor samples and non-small cell lung cancer cell lines. So let's jump into the paper. So figure 1 here shows the expression of c-MET across several cell lines that you see here, okay? This is a Western blot with an antibody against c-MET. And you could see that these various non-small cell lung cancer cell lines express the c-MET protein. Note that there are two major bands, one at 140 kilodaltons and 170 kilodaltons, that are detected by the anti-c-MET antibody. And that is because there is a leader sequence that is cleaved off in c-MET. The leader sequence is common to proteins that are destined for the cell surface or for secretion. This is cleaved off in the endoplasmic reticulum once it is targeted there by the signal recognition particle. Okay, so that's what explains that. What is important here is that they showed that the loading control over here, what they use is a PI3 kinase. And the levels of PI3 kinase are not entirely consistent because it seems like some lanes have less loading than others. However, I would argue here that PI3 kinase is not a good choice of loading control. And the reason is the levels of PI3K can be upregulated or downregulated in cancer. Typically, it's more common to use a housekeeping gene, one that is present in all cell types and at relatively steady levels, independent of the status of the cell, meaning that the levels don't fluctuate much depending on its physiologic or pathophysiologic state. So actin is one example of a gene that is frequently used as a loading control. PI3K is a well-known cancer gene, and its levels can fluctuate across normal and tumor samples and across different tumor types. So that's why I don't think that that was a great choice here. One thing that they note is that cell line H661 does not express c-MET, even though protein was clearly loaded here. Okay, so the other possibility could have been that they maybe just didn't load protein into this lane, but that's proven to be incorrect by this loading control here. The other possibility is that this the antibody may detect an epitope on the c-MET gene that is mutated in this cell line. So that's another possibility, and we don't know that at this point. They also point out that there is significant expression at the protein level that is correlated with the level of the RNA. So they looked at RNA expression using an Affymetrix array. And when the level of the RNA was low, the level of the protein was low. When the level of the RNA was high, the level of the protein was high. And this correlation was pretty good across most of them, except for H661, which did have some RNA expression, but it did not appear to have immunoreactivity at the protein level on the Western blot. They also point out that SKLE1 seems to have higher level of protein expression, but lower level of RNA expression. But here, I do think that there is maybe more protein loading, if you can use this as a marker of higher protein loading compared to adjacent lanes. So that might explain the relative higher levels of protein in this lane compared to this adjacent lane, for example. Okay, so what this whole figure shows in brief is that MET is expressed across various non-small cell lung cancer cell lines, and the level of the RNA seems to correlate overall fairly well with the level of the protein. Now, this figure 1b here is showing that in this particular cell line, A549, that the addition of hepatocyte growth factor, which is the ligand for the MET receptor tyrosine kinase, and for that reason, MET receptor tyrosine kinase is also known as the hepatocyte growth factor receptor. Okay. Or CMET or cellular MET is expressed at these various time points, but hepatocyte growth factor is incubated with these cells for varying lengths of time, up to 1 hour. Okay? And they showed that even at 7.5 minutes after adding hepatocyte growth factors to the cells, phospho-MET is detected, meaning that upon ligand binding, phosphorylation of tyrosine 1003 and these various other tyrosines in the tyrosine kinase domain of PMET, Y1003 is in the juxtamembrane domain, but these phosphorylations happen very quickly, and they are sustained over this period of the study. Okay. There is some phospho-MET that seems to be detected at very low levels, even without the hepatocyte growth factor. Some authors have suggested that these phospho-specific antibodies may not be so phospho-specific, and they may even bind to the unphosphorylated epitopes as well. And that is certainly a likely possibility. The other possibility is also that there might be low level of phosphorylation in the absence of ligand as well. Okay. What we notice with phospho-ERK, which numbers the MAP kinase, and we have two isoforms, ERK1 and ERK2, and they are also rapidly phosphorylated. SK6, which is downstream of the mTOR signaling pathway. So phospho-ERK is downstream of the MAP kinase signaling pathway. SK6 is downstream of the mTOR signaling pathway, or the PI3K kinase signaling pathway. Its phosphorylation is much slower to reach a peak level. Okay. And similar seems to be the case with PDK1, AKT, and of course mTOR. It's harder to assess though, but it seems that MAP kinase signaling takes place fairly rapidly at 7.5 minutes, but it seems to take about an hour for the mTOR signaling pathway to reach a peak. And actually, we don't know if that really is the peak or not, but we assume. And then PI3K levels are the same at each of these time points. So that is the loading control here. Okay. The important take-home point here is that the hepatocyte growth factor receptor, or MET, is expressed and functionally active and responsive to hepatocyte growth factor, and it is able to initiate signaling in the MAP kinase pathway as well as the PI3K AKT mTOR signaling pathway. Okay. And we know that that is the normal physiological role of MET. So this is all in vitro work looking at protein expression and RNA expression. Looking directly at the cells themselves, we see that cells that are grown on cover slip here and C at the top and at the bottom, and after 15 minutes, of incubation with the hepatocyte growth factor, followed by immunofluorescence detection for an anti-MET antibody shows significant upregulation of MET. Okay, so this is a really nice cellular correlate of what is being seen here at the level of protein immunoglobulin. Okay, so that's figure one. Again, nothing extraordinary here except that that MET seems to be an important protein that is expressed in various lung cancer cell lines. Okay. So that was looking at cell lines. It would be nice to know if they're expressed in tumor tissues as well, because the tumor tissues themselves are more are closer to what is happening in the real world in patients, because cell lines can always aberrantly express new proteins that are not really expressed in the physiological pathophysiological states. Here, the immunohistochemical staining with MET antibodies shows expression of MET in various types of lung cancers. Okay, what is important, though, is that does not appear to be expressed in normal lung tissues at any significant level, but at fairly higher levels in these cancers. I will say, I guess one critique that I would offer here is that as a pathologist, I like to see the H&E stained images as well, so I can confirm the histological diagnoses that are given here. Although this does look like an adenocarcinoma, this does seem to be a squamous cell carcinoma, and so on and so forth. Okay, so overall, the morphology on the IHC stain does seem consistent with the morphologic diagnoses given here. Now in B, this is showing the percent of tumors that they analyzed that had activation of MET. So this is just looking at MET. It'd be nice to know whether MET is actually functionally active, and if it's functionally active, it should be phosphorylated both in the juxtamembrane domain and in the tyrosine kinase domain. These various residues are all in the tyrosine kinase domain, and when active, they are phosphorylated. Okay, all of these phosphorylations that you are seeing of these tyrosines as a result of autophosphorylation. Okay, in adenocarcinoma, both juxtamembrane and tyrosine kinase domains are phosphorylated in about 40% of tumors, a somewhat higher percentage in large cell carcinomas, and in small cell lung cancer. What is bizarre, and I don't know exactly how to explain this, is that only the juxtamembrane domain is phosphorylated in squamous and squamous cell carcinomas and lung carcinoids. Okay? So if anyone has an explanation for this, I'd love to hear it in the comments. Okay, so again, here the take-home point is that even in actual tumor samples across many different histologic types, MET is expressed and is functionally active as determined by these phosphorylation status in the tumor samples. So that's actually a really good news for people who are wanting to develop targeted therapies in lung cancer, because this could be a potentially new driver, a targetable driver in lung cancer. Okay? And for pharmaceuticals, this is really, really interesting. Okay, so we know that MET is expressed. We know that MET is active, and it would also be interesting to know what the mechanism of overexpression could be, and what the mechanism of this increased functional activity could also be. Okay, so in some cases, it could be because of increased intratumoral hepatocyte growth factor. That's been shown. It could also, in some cases, be due to tyrosine kinase domain mutations, which has been shown before this paper was published in papillary renal cell carcinomas. Their sequencing of MET, however, revealed a different mechanism of MET activation in mutated samples. So some of these tumor samples showed semaphorin domain mutations that's in the extracellular portion of the MET receptor. Others showed mutations in the juxtamembrane domain, okay? And that was, both of these are unusual. And this particular sample also has a splice variant that resulted in the skipping of exon 14, okay? This was an in-frame alteration, which is unusual in exon skipping events. And for that reason, most of these exon skipping events are actually seen in tumor suppressor genes because they will result in a loss of function because the fusion or the splice alteration is out of frame. Okay, in this case, it's in frame, and presumably it is resulting in a gain of function. We don't know that at this point. Okay. All of these mutations were found in the heterozygous state, with the exception of this splice variant. Okay. And that's consistent with MET being an oncogene. Usually in oncogenes, you only see mutation in one of the two proto-oncogenes. Okay. They also analyzed the adjacent normal to see if this mutation was in the germline, which would be a good thing to do because a small but significant fraction of tyrosine kinase domain mutations in MET and papillary renal cell carcinomas are in the germline, but none of these appear to be in the germline from the ones that were analyzed. There were some that were not analyzed. For the most part, they were heterozygous. When they were present in the adjacent normal tissue, in some, but they were not always present in the adjacent normal tissue, as you can see here. Okay, those are the negative signs. So how do you explain these mutations in adjacent normal, quote unquote, normal, histologically normal tissue? Well, this could be related to a mutation before malignant transformation, okay? So that's what I suspect is going on here. And this really sheds some light in cancer biology that even histologically normal tissues can have some pre-malignant changes. So moving on here, this is looking at MET expression that has been evaluated in various lung cancers, and this is in normal tissues versus adenocarcinomas, lung carcinoids, and small cell lung cancer. They are all significantly overexpressed compared to normal tissues. This is the localization of these various mutations diagrammed on the protein map here. The semaphorin domain encoded by exon 2 is where those mutations are seen. Those are in the extracellular part. So all of this is extracellular. Here's the transmembrane domain. And after that, all of this is intracellular. And that, of course, includes the juxtamembrane domain and the tyrosine kinase domain. Okay. Notice there were no tyrosine kinase domain mutations here. So the mechanism of activating MET in lung cancer seems different than papillary renal cell carcinoma, and that's interesting too. Of course, the juxtamembrane domain is lost or mutated by various mechanisms, and it has been known that this region regulates receptor internalization and subsequent degradation by the proteasome or the lysosome. Okay, and this is all mediated by a phosphocyte 1003, tyrosine white 1003, that is phosphorylated and subsequently bound by C-Cybl, which is a E3 ubiquitin ligase. So when juxtamembrane domain is phosphorylated, which is what happens when the receptor is activated, eventually it'll be bound by C-Cybl, ubiquitinated, and then the receptor will be downregulated. Okay, so it is as important to turn off a signaling pathway as it is to turn it on. And it is suspected, and although again we don't know this yet, at least from this paper, that these receptors should be increasingly stable and overexpressed on the cell surface. This might be one mechanism where the level of the RNA could be low, but the protein could be highly expressed, okay? Because the protein is increasingly stable. Another possibility could be that the RNA is increasingly efficient at being translated by the ribosome, so there's overall more protein synthesis. So the level of the protein could be manufactured at a higher rate. It could be degraded at a slower rate. And both of those could contribute to increased levels overall. Okay? So that is post-translational gene regulation. I didn't talk about these cell lines, but some of these cell lines were wild type for MET. There was one that had a heterozygous mutation here in the juxtamembrane domain, which is encoded by exons 14 and 15 here. Okay. All right. So I will point out one more thing, that this juxtamembrane domain has some homology to EGFR juxtamembrane domain, and that juxtamembrane domain in EGFR is known also to have a C-Cybl binding site and is regulated by C-Cybl through downregulation by ubiquitination. Okay? So at this point, we can suspect that is what might be going on due to these mutations as well. Okay, so far we know cell lines and tumor samples all express MET, and that MET is functionally active. And in some cases, although not most cases, of lung cancer samples, there are mutations. Okay, so I can't remember how many they actually sequenced, but okay, 127 is how many they sequenced, and they only found these handful of samples, like maybe 10 or so that were mutated here. Okay. What is interesting, I don't know, I'll have to go back and see how they sequence these, but there was only one splice variant that was detected here, but generally about 2 to 4% of lung cancer samples will have, and these were lung adenocarcinomas, and 2 to 4% will have splice site mutations. And for a number of reasons that we might discuss in another paper, these are more difficult to detect by DNA sequencing. So let's see how they actually did the sequence analysis here. So, okay, results, functional activation, novel MET alterations. We evaluated lung cancer cell lines and lung adenocarcinomas for the presence of c-MET mutations using DNA sequencing technique described previously, which is not that helpful. Based on our prior experience, the non-small cell lung cancer study was chosen for c-MET mutation in lung adenocarcinoma tissues. Okay. So they analyzed SEMA, juxtamembranes, and the tyrosine kinase domain. So they didn't sequence everything. This may not have been like a next-gen sequencing type of thing. This was in 2005, so it's probably not next-gen sequencing, okay, because that didn't really come into routine, at least clinical use, till 2010. I suspect this was more of a Sanger sequencing type of analysis. especially since they're looking at specific domains here. Okay. Okay, so that is that. And certainly these Sanger sequencing can certainly miss many types of splice variants. And that's probably why they only found one in all of these tumor samples. And we expect that, they should probably detect it anywhere from, 2 to 6 in this many tumor samples. Okay. And I don't know if they confirmed any kind of way. It doesn't seem like they actually confirmed with RT-PCR these splice variants. Okay. So moving on. So we know the distribution of these mutations now, and now they want to examine how. So now they know that MET is expressed. They know that MET is functionally active. They want to evaluate now whether inhibiting MET will result in inhibiting downstream signaling, and ultimately will it result in cancer killing, right? So this study includes Pfizer, and I'm sure that they are especially interested in understanding not just MET biology and the MET mutations, but also whether you can target it. Okay, so let's see what they found. Okay. So small interfering RNAs are about 10 to 20 MERS that are comprised of RNA sequences that are antisense to a mRNA of interest. So siRNA MET here is complementary to MET mRNA, and when it binds to MET mRNA, it results in its degradation. So siRNAs are added or transfected to cell lines in order to knock down the level of a targeted transcript. The idea being that, can you knock down MET and stop downstream hepatocyte growth factor signaling? So let's see if that's the case. Okay, so when neither hepatocyte growth factor or siRNA is added to the cell line, so this is looking just at the cell line, your c-MET, okay, so it's expressed, and we know that it was expressed in A549 cells from before. There's probably some non-specific labeling of phospho-MET here, okay, for reasons I explained before. There's no p-AKT phosphorylation, okay, so the AKT mTOR signaling pathway is not active, okay, and this MET is probably not active either. This is probably some non-specific labeling, unphosphorylated MET. And here they actually use actin as the housekeeping gene for the loading control. Now you add hepatocyte growth factor, you expect MET to be autophosphorylated, and it is, okay? You also expect that AKT signaling would be activated as well, and it is, because AKT is now phosphorylated, okay? And this difference here is not due to more loading, because you see that the actin levels are the same, okay, or similar. Now, what happens if you knock down MET first, and then add hepatocyte growth factor to activate that? Well, MET is knocked down, okay? And that's not due to less loading, because the level of actin is similar as these other two lanes. Phospho-MET is decreased, okay? So in the presence of hepatocyte growth factor, because there's less MET. And more importantly here, you are actually knocking down downstream signaling through phospho-MET and phospho-AKT, okay? Which means that knocking down MET does inhibit downstream signaling in this cell line. Moreover, they looked at cell viability assays, and they show that in these various cell lines, inhibiting with an inhibitor, SU11274, which is a specific tyrosine kinase inhibitor of MET, cell viability goes down, okay, in a concentration-dependent manner, okay? So you can shut down the pathway, and shutting down the pathway seems to lead to the inhibition of cell growth, okay, in vitro, in these cell lines, okay? And here is looking at two different cell lines with this inhibitor. They do mention that they looked at cell viability with siRNAs too, and they found similar results. Okay, now looking at these two different cell lines, you see that this one, the wild type, in the absence of inhibitor and growth factor, okay, there is no phospho-MET, okay, or very little. No phosphorylation of S6 kinase, which is downstream of mTOR. No phospho-ACT, again, downstream of mTOR. No phospho-ERK 1 and 2, which is downstream of which is the MAP kinase, of course. And c-MET levels and beta-actin levels are similar in all of these lanes, okay? So differences in loading don't explain the differences you see here. Okay. Now, if you add hepatocyte growth factor, you do see increase in downstream signaling. But once you add inhibitor, okay, and you add, even in the presence of hepatocyte growth factor, there is at least decreased phospho-MET and decreased phospho S6K at ERK signaling, okay? So that's what we're seeing here compared to here. Okay, there is some decrease. You didn't completely shut it down, but we did reduce it. And the pattern here for H1993 is a little bit different. There does seem to be more phospho-MET that can't really be fully explained just by nonspecific binding, okay, even in the absence of hepatocyte growth factor. So does H1993 have a mutation in MET? Well, we don't know, okay? That results in activation of MET independent of ligand. Okay? But interestingly enough, regardless of whether hepatocyte growth factor is present or not, you are able to down-regulate phospho-MET signaling in each of these cases, okay? Compared to the absence of the inhibitor. Okay. So all of these results are very interesting. They're all done in vitro, and it all points towards MET being an interesting target for non-small cell lung cancer. Okay. I think there's more work that needs to be done here. So, for example, all of this work was done in cell lines. Can any of this be done in a more in vivo situation in, for example, mouse models of non-small cell lung cancer harboring MET overexpression or MET mutations? Are there specific MET mutations that might be more sensitive or less sensitive? And then what is the efficacy of these drugs and their toxicity in humans? So lots more work to be done, but this was a very good initial characterization of MET in non-small cell lung cancer cell lines and in non-small cell lung cancer tumor samples. And more work will be done, and we'll present another paper that follows this one that better characterizes some of these mutations. Thank you for now, and I appreciate your time. If you liked this video, please hit like and subscribe to my channel. Thank you.